Welcome to this particular lecture on isolation and characterization of E. coli. In this particular lecture, myself and my colleague Dr. Sudeshna Shamchaudhuri will be discussing the different aspects of E. coli and its discovery and also an experiment to denote its isolation and characterization. The systematic position of E. coli or Escherichia coli belongs to kingdom Eubacteria, phylum is Proteobacteria, class Gamma Proteobacteria, order is Enterobacterialis, family Enterobacteriaceae, the genus is Escherichia and the species is coli. This particular bacteria was denoted by Theodore Escherichia way back in the year 1885, but at that time he named it as Bacteria coli because it was isolated from colon. Subsequently, it was Migula who actually showed that this particular bacteria is rod-shaped. So he named it as Bacillus coli. But it was Castellani and Cammers in the year 1919 who gave the modern name Escherichia coli following Escherichia. But this particular bacteria actually was the tool for different biotechnological experiment, and one such particular tool was denoted by Lederberg and Tatum, who denoted the phenomenon of sexual reproduction by conjugation in this E. coli, because there were the pili, and this pili is acting as a conjugation bridge which is helping in the transfer of the extra nuclear plasmid. Subsequently, it was Benzier who in the year 1955 denoted the different types of gene structure, and not only that, he actually showed that these genes are present in a linear fashion. So in that way it indicated the linear chromosome of E. coli. But it was Cohen and Boyer in the year 1972 who actually used E. coli and produced the recombinant E. coli using the restriction enzyme. But this bacteria, when we come to their cell structure, is actually not a pure rod. It is neither rod nor round, so it is called a coccobacillus, and it is having peritrichous flagella, that is flagella all around. The genome is very specific. The genome of E. coli K12 was denoted, and it was found out that this particular bacteria had 46,39,221 base pair, and this is also having 4,288 cistron, about 2,584 operon, about 7 rRNA, and the coding distance, which is so important, is only 118 base pair. So that means the frequency of the functional gene is quite high, and this actually makes them a perfect inhabitant in the intestine. This is the intestinal wall where the villi and this bacteria is trapped by the villi. So they remain within the intestine very nicely protected by the villi network. And at the same time, they can produce some toxin, which is Shiga toxin, and Shiga toxin actually is causing food poisoning. So very often when we see that people having some food contaminated with E. coli, they may show this poisoning of Shiga toxin. And this is the basic binding of this Shiga toxin. This is the Shiga toxin producing strain, which is binding with the TIR receptor, and it is producing the toxin. This toxin gradually enters inside and it causes the basic immune response in the blood cell. It will prevent protein synthesis. Different toxic implications will be there, which may also include the bleeding from the intestinal wall. So it is enterohemorrhagic in nature. This particular bacteria can produce vitamin or vitamin K, to be specific, from the chorismic acid pathway. The dihydroxy naphthanoic acid is the intermediate, and this will ultimately produce the menaquinone and phylloquinone, and this vitamin K production make them unique and make them survive in the intestinal mesh, where by outnumbering the other pathogenic organism. So very often when the E. coli, which is non-pathogenic, may remain inside the intestine and produce vitamin K for our own benefit. We come to their different types of reproduction. As I have already said, that conjugation is there, but at the same time, they also reproduce most commonly by binary fission, which is simply a mitosis, that is the nucleoid will divide and followed by the cell division. At the same time, they produce the conjugation. You can see the sex pili through which this extra nuclear plasmid or the F plus plasmid is transmitted. So you can see that the F plus is donating the F plus plasmid and the F minus becomes F plus. So this is very common, and sometimes the F plus may get into the nucleoid and producing the high frequency recombinant or HFR strain. Now coming to their growth, this particular bacteria ideally grows in nutrient agar medium. Of course, the temperature ranges between 37 to 49 degrees centigrade, and you can see these are the discrete colonies which are translucent balls growing inside this Na agar plate, and oxidized trimethylamine oxide is reduced and producing trimethylamine. So production of trimethylamine from trimethylamine oxide by removing the oxygen is very important test for this particular bacteria. But this is really a matter of controversy whether it is pathogenic or non-pathogenic. Actually, it is a diverse organism. All the strains are non-pathogenic. The E. coli K12 is non-pathogenic, which is not producing toxin, which is not having any specific invasive action, but at the same time there may be pathogenic, which will produce the toxin, which will have the capsule, which will have the adenosine, which will have enterohemorrhagic property, and these properties actually are donated from this non-pathogenic strain. So this brings us into a story where from a non-pathogenic strain will become pathogenic, and thereby we can say apparently we were not harmful gradually becomes harmful. So these are some of these serogroups. Now when we come to that, we must understand these serogroups are created on the basis of the surface antigen. This surface antigen can be the O antigen present in the lipopolysaccharide layer. It can be the H antigen associated with the flagella. It can be the K antigen associated with the capsule. Accordingly, we give different nomenclature. Say for example, O157KH7 is a very common pathogenic strain. And this pathogenic strain ultimately may produce different types of infection or may produce other serogroups as well. Accordingly, there may be 12 major serogroups, but actually speaking, there may be 190 such serostrain in E. coli. So, E. coli modification is very common.
Sir, I have a question. What is the importance of these serogroups?
Serogroups are created because of the natural mutability of the bacteria. Now, why do they do it? Say, a particular bacteria which is being treated by antibiotic is getting mutated, producing a new serogroup. So this new strain will not be affected by the antibiotic. And that is the reason why you understand E. coli is a very common source of urinary tract infection. And you know, UTI causing bacteria, say one antibiotic is acting in the morning, in the same evening you will find that antibiotic is not working. So the serogroups mainly, they help in pathogenicity and of course better adaptability in the human environment. Now, as I was telling that this properties keeps on changing, but there is a typical neotype which is O1K1H7 may be X-gal positive, they may be indole positive, and these are basic tests by which we can identify that particular bacteria. O157H7 strain is producing adenosine. Now adenosine production is a very important tool. Why? Because this adenosine will help in the attachment with the villi, and they will help in the attachment of the individual bacterial cell. In other words, it will help in the colonization of the bacteria in the intestine. And of course, there may be pathogenicity, may not be, depending upon other properties of that bacteria. If it is pathogenic, it will not remain only in the intestine, because from the intestine it will get into the bloodstream, and then it will cross the blood-brain barrier and it will affect the meninges, and it causes the neonatal meningitis, which is a fatal disease. And this swelling of the meninges is actually causing tremendous pressure on the brain itself, and thereby the CSF cannot grow, the brain cannot grow, so the child may suffer and the child may die also. Now this is the basic colonization or the pathogenicity of the E. coli inside the intestinal endothelium. They will show attachment, they will invade, they will replicate, they will colonize, and ultimately get into the deeper tissue. and they may be carried by the macrophages. They will ultimately get into immune system and will produce hypersensitive reaction to the production of these interleukins and other molecules. So it is a complicated case the moment it comes to blood. Now this particular strain, enterohemorrhagic E. coli, actually comes mainly from cattle, and the moment it is coming inside the human intestine, it will develop specific symptoms like stomachache, diarrhea, fever, vomiting within 3 to 8 days. So the source is food, and the toxin comes into the intestine, and there may be the hemolytic uremic syndrome or HUS, and there may be death in 5% cases. So this is a typical case of EHAC. Of course, there is another very important strain, it is called commonly UPEC, that is uropathogenic E. coli. This particular bacteria commonly is there in the colon or in the rectum. It goes into the urinary tract and will cause the urinary tract infection. And that sometimes they can go up to the kidney and will cause hydronephritis, which can be in the long run produce renal failure. They produce different recombinant strain of E. coli, where you can see the gene for the production of insulin was inserted in E. coli, the work of Cohen and Boyer, and they expressed it in E. coli, and now the E. coli is producing insulin. So this is called recombinant insulin, and naturally the price of insulin came down after this discovery. Of course, this is a remarkable discovery. Apart from that, there are some specific ways by which E. coli can accept different genes. Say for example, from Campylobacter, it will take some gene with which they will ultimately produce the outer LPS layer shorter, that is the trimming of the LPS layer, and thereby they will be more stable in human gut. Now basically this is in non-pathogenic strain because they are friendly. If they grow, then there will be more vitamin K.
Sir, how is the expression of this transglycosylase system in E. coli bringing about its stability in the gut?
You see, whenever the external growth of the glycosylation is more, naturally the more receptor molecules are there. If the more receptor molecule, there will be more interleukin, more hypersensitivity, more allergic reaction, more complication. So naturally, when they are having a trimmed outer LPS layer, naturally the disturbance will be less because they can evade all these reactions. There are different other bacteria having some genetic segments attached. Whenever that particular bacteria is remaining in human gut, along with other microflora, they have taken up certain gene and they have actually internalized those genes and they have utilized it. So basically, this gives a definite idea that all the good genes of different other bacteria might get into E. coli, and this might have given good properties of E. coli which are non-pathogenic to us. So that is making E. coli a very good helpful microorganism in our gut. These genes may come from extranuclear plasmid, it may come from fudge, it may come from other bacteria, and they have all been expressed in a single genome, and depending upon the expression of these genes, they can be toxic or non-toxic. So basically we can now call it the Escherichia which we get now is not that original Escherichia. This Escherichia is a mixture of all these genes of different other bacteria or bacteriophage. And definitely when I say bacteriophage, definitely there will be a viral pathogenicity island which will induce pathogenicity in this particular bacteria. On the basis of their growth and development and transformation, the Hamiltonian problem can be solved using this bacteria as a computer. So we can call it a bacterial computer solving a complex mathematical problem. So these are certain greater uses of E. coli apart from their basic utilization in the human gut.
E. coli is normal bowel flora of different mammalian species. It is found as indicator organism in contaminated water. It may be pathogenic or may not be pathogenic. If it is pathogenic, it is due to the presence of some virulence factor. What are they? Enterotoxin, virotoxin, type 1 pili, as well as the motility factor and antibiotic resistance. First one, collection of sample. Requisite specific site must be selected according to the sampling and analysis plan SAP, and the representative sample must be collected. It will be collected in some satellite autoclaved bottle. The bottle will be labeled and it will be replicated in number. In 250 ml of satellite bottle, 200 ml of water will be collected. The lid should be tightly capped and shaked well for the homogenization of the solution. After collection, the sample must be preserved below 10 degrees centigrade inside the refrigerator. There will be not more than 2 hours in between collection and analysis.
Ma'am, I have a question. How do we select a sampling site on the basis of the sampling and analysis plan?
Basically, SAP or sampling and analysis plan is based on the biostatistical analysis, because the contaminated water with microorganism is not homogeneous solution. To make it non-randomized, so it is collected from different sites at different times. So that is the sampling and analysis plan. It includes MacConkey agar, EMB agar or eosin methylene blue agar, gram stains including crystal violet as well as safranin, lugal iodine, TSI agar, triple sugar iron agar, 5 basic sugars, dextrose, maltose, lactose, sucrose as well as mannitol, Daham's tube, 3% H2O2 for catalase assay, methyl red, Kovacs reagent including alpha naphthol as well as 40% KOH, Barrett's reagent, and apart from which nutrient broth and peptone water broth with tryptophan will also be taken. First, the primary culture that will be made by inoculation of the collected sample within the nutrient broth, and that will be kept at 37 degrees centigrade for 18 to 24 hours. After 24 hours, the growth will be streaked over the EMB agar as well as over the MacConkey agar. Then, after getting characteristic morphology of the colony according to the size, shape, opacity, color, elevation, surface texture, age, then that will be subcultured over EMB agar and MacConkey agar, and then that will be repeatedly subcultured to get the pure culture for the homogeneous colony. But at every stage, we have to have the standard culture of E.Coli obtained from the MTCC. The cultured sample will be taken for the microscopic study or gram staining to ensure whether it is E.Coli or not. Usually, E.Coli is pink in color, small rod-shaped, gram-negative bacilli. To distinguish in between the motile and non-motile bacteria, the liquid culture will be taken over the hanging drop slide, that is also called as the curved slide, and that will be observed under 100x power objective. That will actually help to elucidate whether the organism is motile or non-motile. The streaking over TSI agar, triple sugar iron agar, that will actually help to ensure whether the organism is able to produce H2S or not. That can be observed by the small black precipitation. That I have already told you that the sugars, basic 5 sugars will be taken like dextrose, maltose, sucrose, mannitol, lactose, etc. So 0.2 ml of the culture will be taken and all sugars will be incubated, and after some times, after 37 degrees centigrade 24-hour incubation, you can follow the acid production that can be followed by the change in color of the broth, and the gas production can be followed within the inverted Daham's tube by the bubble production. 3 ml of catalase reagent, that is 3% H2O2 will be taken inside the test tube, and with glass rod, some single colony will be taken from the pure culture and will be merged inside the test tube. So you can follow the effervescence of the oxygen. If catalase is present, that will help to disintegrate H2O2 to H2O as well as oxygen. Within the 5 ml of methyl red Voges per square broth, you have to inoculate the collected sample, and 5 days incubation time will be given. After that, 5 drops of methyl red indicator will be added. As we know that E. coli follow the mixed acid fermentation pathway, they will produce enough acid. So pH will be lowered at pH 4. So the color of the methyl red will be red enough to show the positive result. So here once again, within the 3 ml of MRVP broth, the sample will be inoculated. And after that, after some incubation time, you have to give Barrett's reagent, that is 0.6 ml 5% alpha naphthol as well as 0.2 ml of what? Potassium hydroxide, KOH, 40% KOH. So that will actually help to mix with the solution and allow to stand, and after that you will have the pink or purple red color formation showing the positive Voges-Prosker test. But here you can follow the negative Voges-Prosker test result.
Ma'am, what is the principle of this Voges-Prosker test?
Actually, E. coli is fecal coliform. They follow the mixed acid fermentation pathway. But Enterobacter erogens is the non-fecal coliform. They follow 2,3-butanediol pathway, and ultimately it will produce 2,3-butanediol and acetoin. These acetoin, with the help of alpha naphthol and 40% KOH oxidizing agent, is getting converted to acetoin. These acetoin along with guanidium group of the peptone give the red quinonoid coloration, but that is actually absent in case of E. coli. 3 ml of the liquid culture is taken. We are adding first 1 ml of diethyl ether. So we will wait for some time so the ether level will rises at the top. So it will make the organic layer. After that, we have to add the COVAX reagent. So COVAX reagent contain para-dimethyl aminobenzaldehyde. And when we are adding this COVAX reagent, we will shake it and we will actually stand them for 5 to 10 minutes. Then you can follow the presence of the red ring in between the ether and the medium. So that will indicate the positive test for the indole. It is positive for the E. coli. E. coli is able to secrete out tryptophanase. So this particular broth contains 3 ml peptone water broth along with tryptophan. If tryptophanase is getting secreted out, that will be disintegrated and indole will be produced. That particular indole along with COVAX reagent will be able to make this color, red color or purple color ring. E. coli in EMB agar is able to produce small black centered colony with green metallic sheen. That is the typical colony for the fecal coliform organism. So here also you can also observe how beautiful metallic sheen is actually observed. In MacConkey agar you can follow bright pink colonies as well. So bright pink colonies are very well manifested here also. So that is the subculture result, and the gram staining result of E. coli will show gram negative small rod shaped bacilli. Next one, so motility test on the hanging drop slide. By hanging drop technique, you can observe the motile bacteria that will confirm yes, it is E. coli. Sugar fermentation, that I have already told you that it follows the mixed acid fermentation pathway. So by fermenting all those 5 sugars, it is able to form acid as well as the gas formation can be observed as bubble within the inverted Durham's tube. But you know that there are different types of sources of contaminated water, okay, drain water, pond water, etc. So their ability to ferment sucrose, mannitol or different sugars are quite different, but somehow they are able to ferment lactose, okay. So they give the mixed acid like lactic acid, acetic acid, formic acid, propionic acid, all organic acid together. From this table you can follow the indole test is positive for E. coli. You can observe it by the formation of the red ring. Methyl red test is also positive because it shows the red color. Voges-Prosker test is negative, okay, because it does not follow the 2,3-butanediol pathway. It follows what? Mixed acid fermentation pathway. Catalase test is positive because bubble formation you can observe. TSI agar, very interesting. So in case of TSI agar, the streak or stab is made, right? So you can follow the yellow slant or yellow butt, okay, as well as you can follow the gas or bubble formation, but you cannot follow the black precipitation by H2S production. So that is also the confirmatory test of E. coli on TSI agar. You know that for particular experiments some precautions should be taken. First one is that the sample will be collected from the 15.2 centimeter depth, and the surface scum should be avoided. And when we are collecting the sample, you should not touch the inside of the bottle because it is autoclaved. And the lid should be tightly capped. It will be shaked well for the homogenization of the solution or to get the homogenized solution. And the standard culture should be taken to compare each and every step. So that's all about the precautions that should be taken during the course of experiments.
So, in this particular lecture, we have learnt about the different introductory aspects of E. coli, and along with that, we have learnt an experiment how to isolate, identify and characterize E. coli. Thank you.
Thank you.